Thank you for joining me on UCSD Guestbook. Tonight it is my pleasure to welcome Chuck Stevens, who is professor and investigator of the Howard Hughes Medical Institute at the Salk Institute here in La Jolla. Welcome, Chuck.
Thank you, Nick.
Chuck is a neuroscientist's neuroscientist. He has a keen eye for important scientific problems and a wonderful ability to solve them with both imagination and elegance. The problems on which you have worked, Chuck, are at the core of our understanding of how the brain works. Electrical excitability, the production of action potentials, transmission of signals from one nerve cell to the other at synapses, and changes, long-term changes, in the properties of these synapses that are the connections among nerve cells. Let me start, if I may, by asking you about one of the most astounding results of your work, and that is the discovery that most synapses in the cerebral cortex are unreliable, that an electrical signal, an action potential arriving at the end of one nerve cell fails to release, very often, fails to release transmitter molecules that signal to the next cell. If we were to build a computer on this principle, we would be very unhappy with the results. Is there some hidden benefit of this property of the nervous system?
Yes, I believe so. I think it probably is a consequence of how we have to have our nervous systems in order to be fault tolerant. We have a lot of nerve cells up there, about 100 billion, and if we depended on every single one of them, they're bound to die and our computer would stop working. So what we have to do is we have to make a computer that still works no matter which cells you take out, even if you take out quite a few of them. And in that case, you have to have redundancy. You have to have many cells doing the same thing. And that means that you have to pack a lot of cellular components in very close together. And to do that, they have to be very small. And to be very small, then you have to conserve on your resources. And one of the ways of conserving on resources is to make the synapses be unreliable. But they're not really unreliable. They're perfectly predictable. It's just they're unreliable in the same sense a coin is. So if you flip a coin any 100 times, you're pretty sure you're going to get very close to 50 heads. But you never know on any flip whether you're going to get a head or a tail, and synapses are like that. Mm-hmm. They are quite reliable on the average and unreliable for any given time. But it has a number of advantages. One of the advantages is if you have a synapse that only transmits its signal on, say, 1 time out of 10, if you increase it to 2 times out of 10, you've doubled the strength of that synapse. And if you increase it to 10 times out of 10, you've made the synapse 10 times stronger, because the average transmission would be 10 times bigger. And so the nervous system makes use constantly of this unreliability by making it go up and down in order to adjust dynamically the strength of the synapses, how effective they are at influencing the cell, their target cell. Mm-hmm.
This leads naturally to another very important area of your work that is focused on the mechanisms underlying what's often referred to in neuroscience as long-term potentiation, the long-term strengthening of synapses. And a question that comes up in this context that you've just developed for us is the molecular mechanisms by which this is achieved. What are the current understanding? What is the current understanding of this process?
Well, over the last 15 years, there has been a lot of effort devoted toward understanding the long-term changes at a synapse. And long-term, in this case, means sort of the length of an experiment, which is sort of a half an hour or an hour. And those are the easiest things to study. Some things we do know about, a lot of properties, and a lot of things we just don't know. And so at this point, there really isn't any consensus about what the mechanisms are although we know enough about it that we can make use of our information to go on. And so a lot of the focus these days has moved past what's called long-term potentiation to what some people call LLTP, long-term potentiation, which are the mechanisms that would be more likely to be used for storing memories for more than half an hour, for days or weeks. And so that's the current focus of a lot of work. And there, we still don't know what the answer is. We don't know what the mechanisms are. But people believe that it's a change in the structure of synapses or making new synapses. So there's a lot of effort now devoted toward determining how that could happen.
Would something like that involve protein synthesis, the synthesis and insertion of new molecules in the synapse?
Yes. So what happens presumably is that when a memory is stored for more than minutes, for hours and days, presumably you actually change the structure of your nervous system. You actually synthesize and insert new molecules. You make synapses larger. You maybe make more of them. You take synapses away. So it's a real structural change in the nervous system that would be the basis for long-term memories.
It's fascinating to begin to come to grips with the way in which these important processes of learning and memory are actually taking place. Another fundamental result of your work has been the discovery that the nervous system is inherently noisy, that there are electrical signals, much like the noise on a radio station or a TV program, that you showed originate at the synapse, at the site of connection between cells. But intuitively, looking at this property, one might think this would distort or reduce the information transfer in the nervous system. Is there, again, a benefit of this process to the transfer of information in the brain?
Well, to a certain extent, the noise is inevitable because nerve cells are combining information. Each nerve cell receives information from about 10,000 other nerve cells. And this information comes in little pulses that come sort of irregularly. And when you add up pulses from 10,000 different sources kind of irregularly, you get noise, and you just can't beat that. And so what the nervous system has done is it's figured out ways of getting around this noise by averaging over many synapses, by averaging over long periods of time. But it also may be that the noise itself is useful by making sure that the nervous system doesn't get stuck in kind of a regularity that would be bad for it.
It's, again, nice to see that these biophysical properties that initially look somewhat disappointing in terms of the function of the brain, actually, as your work has shown, are significant. In quite another area of your work that you have told us about recently, you've analyzed mechanisms that govern both the size of the brain and the complexity of the brain with a view to understanding how this has changed during the brain's evolution. And this appears to be, on the face of it, somewhat different from your interest in electrical excitability, the function of synapses. How did you become interested in this class of problems?
Well, I've always been a sort of closet theorist, and I always try to understand things in a broad range of things. And over the last 15 years, I've gone away each summer and spent sort of a mini sabbatical, and spent a couple of months working on theoretical issues. And part of the time I'm just learning new things that I hadn't learned before, learning about the cortex, which I didn't know much about at one point. As you do this, you run across interesting puzzles. And in trying to illuminate these puzzles, you sometimes can gain deeper insights into how the brain might be functioning. And so the particular thing you were talking about maybe it gives us a deep insight into how the cortex works, because it seems like all parts of the cortex, the covering of the brain, where much of the computation goes on, it seems like most parts seem to behave very similar to the one, to the part of the brain that we understand the best, which is the primary visual cortex. So some people believe that each part of the cortex is different. Other people believe that the cortex basically uses one computational style.
That it's modular in some sense?
Yes, and that if you understood how any one part of the cortex works, you'd understand in essence how every part works. Of course, there are going to be differences having to do with what goes in and where the output goes to, but a lot of people believe that there's just one computation. And this work that I was doing doesn't prove it certainly, but it supports that idea, and it gives you clues about how information is represented in the cortex, so that it might form as a basis for the computation that's done.
I thought one interesting result that you obtained was that, in one sense, very simple geometrical considerations informed us a great deal about how the structures of the brain had grown and how they were organized. Could you say just a little bit about that? I think it's a fascinating result.
Well, the idea is that the cortex, which is where most of the computations are carried out, over evolution, over primate evolution, so from sort of small monkeys up to us, has grown disproportionately. And you want to know why you would have more cortex, more nerve cells in the cortex per every nerve cell feeding it information in us than you would in a chimpanzee or a small monkey. And it seems to be a result of the fact that the nervous system is taking this information and deriving something from it, and then displaying that thing that it derives on the surface of the cortex. So you not only have sort of map of the visual space, for example, in the visual cortex, but you also have features about that map that are each one displayed on the surface of the cortex. And the hope is that maybe some similar organization could be shown to be true for all areas of cortex.
So this points us in, it really shapes, I think, an important direction for our future work.
If it's right, and we hope so, yeah.
Very nice. Often in science, progress is driven off of the development of new techniques, new methods, new hardware, or more recently, of course, new ways of analyzing the data with new software. How have various technological advances been useful to you in your discoveries?
Oh, well, I mean, not just me, but everybody. And we do experiments every day now that were literally inconceivable 10, 15 years ago. And it's been a combination of modern molecular biology, the ability to make animals that are missing or have modified any gene you choose, the new microscopy techniques so that you can see the features of living tissue much better than you ever could before, computers which have permitted you to process images very quickly so that you can use imaging for studying living tissues, electrophysiological techniques so you can record the electrical activity of the nervous system in ways that couldn't be done before. And every one of these things didn't exist 15 years ago, and now it's just the most standard thing. It's remarkable.
My impression is that, at least in some cases, one can actually program the computer to run a part of the experiment and to go out and have a cup of coffee while the hard work is being done.
Yes. No, the hard work comes when you analyze all the data.
Very good.
But it's certainly true that everyone uses computers for gathering all their data now, and if it's a routine thing that has to be done repetitively over a long period of time, the computer does it, and you can go away and come back after it's finished.
Yeah. Yes. The 19th century polymath, Sir Darcy Wentworth Thompson, in his very influential book on growth and form, stated that numerical precision is at the very soul of science. And Lord Kelvin, writing at about the same time in a similar vein, noted that if one cannot express one's results in numbers, one's knowledge is, if I remember correctly, of a meager and unsatisfactory kind. So I want to ask, Chuck, what you see as the prospects for a quantitative and, in Lord Kelvin's words, satisfactory neuroscience are likely to be in the 21st century with some of these new methodologies and new ways of approaching the nervous system?
Yeah. Well, I think that that it's quite likely that we're going to make a lot of progress in theoretical neurobiology in this century, and I think the reason is that people believe that we really need to have theory to guide our experiments, and usually the theories are quantitative theories. So we have gathered enough background information now that many people believe that it's a time we can start effectively having theories. Mm-hmm. And there are sort of two classes of theories. One class are basically treating biological systems as if they were physical systems, because they are, in a sense. Some of the molecules follow physical laws, and so the natural way to treat them is with physical theories or mathematical theories. But the other thing is that that in order to understand how the nervous system works, we may need to make use of the mathematical techniques that have been derived in physics, and so since physics is the most advanced of the theoretical subjects, that you need to look to them to find the modes of dealing with problems. And I think that there's a lot of people who are doing this now.
Chuck, this reminds me that you have a second life as a theoretical physicist, in fact, and I'd love to hear how, after you got well launched in neuroscience, you found your way into this related but somewhat different discipline, and how your interest in theoretical physics has informed and shapes your approaches to neuroscience.
Yeah. Well, you know, I'm a theoretical physicist, sort of like a lot of people are violinists.
No. You published a book, a textbook on this subject, so I think that qualifies you in spades.
I may, in some areas, be a fairly good amateur, but it's a long ways to be a professional. But it started really because I used to work in biophysics, and there. You need to understand the physical principles in order to understand the biology. There's a long tradition of applying physics to biological processes in biophysics in the nervous system. Mm-hmm. So how the nerve impulse is generated, the mechanisms for that is basically a biophysical problem which depends on physics. And so initially it was for those reasons, because of the work in biophysics. But then over the years I started using the mathematical methods of physics to deal with biological problems that weren't basically physical problems, but I just used the techniques, and I've been doing that more over the
very productively. I would
the last few years. Yeah.
Very, very nice. You have over the years been tremendously successful in writing, either in the pages of Nature magazine or in the pages of Current Biology, articles that take an important finding, not your own, and distill it and make it available, make it accessible to a broad audience. How did you come to start doing this, and what are the features of it that you most enjoy? Terribly useful.
Well, the reason I started doing it was because I was invited by Nature to write a News and Views many years ago, and I believe the first one I wrote was on some work that Steve Heinemann, who's my next door neighbor at the Salk Institute, did early work on the acetylcholine receptor, and I enjoyed it enormously, because it's the kind of writing you do when you're being a reporter is a lot more fun than when you're writing scientific things. You can write with a little different style. You can say things that are funny. You can try to find ways that are interesting to put things, and I enjoy that kind of writing a lot. So I've done a lot of it over the last 10 or 15 years.
You do it marvelously, and the sense of humor that you bring out of it, I think, is a special feature of it, Chuck. It's just wonderful. how have the questions in neuroscience changed over the course of your career? You've been almost 4 decades now at the bench and at the thinking theoretical planes. How are the problems evolving over that time?
Well, it's interesting. It's been driven a lot by techniques, because the techniques have evolved over my career, and that's driven a lot of it. But we now are able to ask questions at the more fundamental level. So in the beginning of my career, one wanted to know which part of the brain might be involved in something, and so now we'd want to know exactly how that part of the brain is involved in it, and maybe even which genes and molecules are involved in it. And the same thing goes for understanding things like the mechanism of the action potential. It used to be just very descriptive, the ways it worked. We didn't understand mechanisms, and as new techniques have become available, we've gone deeper and deeper into understanding, and so now we know the atomic structure of these proteins that are used for generating nerve impulses, and the questions have to do with how these molecules move around in order to make it work, whereas before we didn't even know there was a molecule, we just knew something was changing. Yes. So it's been more and more cellular mechanisms, more and more molecular mechanisms, and at the other end, since we've learned a lot more about the structure and function of the nervous system, bigger questions about how we perceive things, for example.
Well, this leads to 2 questions. I think one is what your sense is of the sequencing of the human and other genomes, what the impact of those events will be on research in the nervous system, our understanding of the nervous system. How do you see that unfolding?
Oh, I think that not just neurobiology, but the whole world has changed. And we don't know how it's going to be changed by the genome yet, and there are a lot of ways we can guess. I think it's going to be like the internet, that the ways that and computers, that the ways that we thought computers were going to be useful are not their most useful thing. I mean, whoever thought that we would be using them for word processing in the days when they used to cost millions of dollars each? And the internet's the same thing. No one could foresee how it was going to evolve, and I think the genome is going to be the same thing. But it's already having just enormous consequences. In the same issue of Science that you just recently had an article, there was another article on a gene, the existence of a gene for tone deafness. Yeah. Where there are people who just can't hear intervals. They're perfectly good in every other way, but there's some gene that's doing this. Yeah. And there are going to be a lot of genes that are doing a lot of things like that. So at the high end, we're going to be figuring out what those genes are for, and having the genome means that you don't have to clone every gene, you just have to figure out which one is doing the job you're interested in at the more cellular molecular level. It's really going to make a difference.
Yes, I think we'll push perhaps more in the direction of functional studies.
Yes.
The discipline that you're pursuing.
It's not the end, having the genome, it's just the very beginning. And really, it's the fact that everyone thought, oh, the human genome, that's wonderful, then we can stop. But now everyone realizes that we need the mouse genome, the rat genome, the dog genome, the chimpanzee genome, the gorilla genome, plus all the bacterial genomes, plus the frog genome, and so we'll live to see, I guess there are a couple of dozen genomes if you include the bacterial ones done now, and we'll live to see hundreds of them.
Yes, yes. Perhaps a quick question about perception, and certainly one of the issues that your colleague Francis Crick at the Salk Institute has been very interested in is consciousness. What's your sense of our understanding of this process, Chuck? When will we have some physical appreciation of how we are either conscious or unconscious?
Yeah. That's very interesting, because before Francis, it wasn't really even polite to discuss questions, for a scientist to discuss questions of consciousness, and now it's been quite a growth industry. Yeah. I think that sort of like cancer, we're not going to understand consciousness all at once. that we're going to be able to understand bits and snatches of it as we go along, and we'll be able to find out certain correlates of things that are conscious, and we will understand those, and maybe not so long in the future, and other things we won't understand. But still, there's the problem of qualia, which I find very, I don't understand how we're going to understand that. Why it is red appears red, or why the blue cups have the nice blue look to them that I experience, and I can't even imagine how we're going to understand that.
Yes, yes, very challenging. Often ideas come to one in unusual ways or in unusual locations. Where do you get your best ideas?
The way I work is by total immersion. So what I do is I start thinking, I have an idea that there's going to be something interesting in a given region of intellectual space. And so I don't even know quite what the question is and what the answer is. And so I just start poking around there, and I poke and poke and poke at it until a question emerges, and an answer. They sort of kind of come together oftentimes. And where it happens is wherever it is that I'm permitted total immersion for the length of time that goes on, and when it happens. Usually happens, oftentimes the idea comes first thing in the morning. So I'll go to sleep at night. I won't sleep very well, and my mind will be grinding away at this problem all night, and then in the morning I'll wake up and I'll know an answer to some small little problem.
Very nice, very nice. One last question, Chuck, I have to ask. If you were granted by some, let's imagine, almighty power, the answer to one question, what question would you ask?
Oh my goodness. Well, you know, I think I probably would be like Francis. I would probably like to understand what consciousness was.
Very important issue, and I hope we can both see that in the years ahead. Thank you for joining me on UCSD Guestbook. My guest this evening has been Charles F. Stevens, professor and investigator of the Howard Hughes Medical Institute here in La Jolla at the Salk Institute. Thank you and good night.